Thank you. I'm going to be talking today about some of our group's recent work developing a platform for targeted transfection of the cerebral vasculature without detectable blood-brain barrier opening, which we feel is an attractive approach to gene therapy for stroke, which is already characterized by a high degree of blood-brain barrier dysfunction, structural instability, and inflammation. So our approach differs from more conventional BBB opening regimes in two key ways. The first of these is that we utilize a cationic lipid-shelled microbubble to which we can directly conjugate our plasmid DNA of interest. This allows for co-localization of the plasmid DNA with the microbubbles, which we've shown in previous studies to enhance transfection in the FUS-targeted region. Additionally, we're utilizing a lower peak negative pressure of pulsing, which we hope will allow for endothelial cell activation and sonoporation of endothelial cell membranes without disruption of tight junctions. And this is building off of some recent work by the Zhang and Villanueva groups, who have demonstrated in vitro that you can sonoporate endothelial cell membranes with FUS and microbubbles. This is an analysis of the extent of blood-brain barrier disruption across a range of peak negative pressures, with more extensive BBB disruption at the higher pressures. As we decrease the pressure, we see decreased contrast agent extravasation on T1-weighted MRI. And at 0.1 megapascal, we cease to see any detectable blood-brain barrier disruption. And it's worth noting the difference between these cationic lipid-shelled microbubbles with more conventional albumin-shelled microbubbles, which you can see at the bottom. Both brains were treated with the same dose of bubbles at 0.4 megapascals, and there's much more extensive Evans blue extravasation in the brain treated with the cationic microbubbles, indicating more reactive bubbles for the same pressure, which is why you'll see perhaps more extensive blood-brain barrier disruption at these low pressures than you might expect from commercially available bubbles. So we use these cationic bubbles to deliver a transgene for M-cherry, and then quantified the vascular selectivity of this transfection by looking at the overlap between that transgene expression and a BSI-lectin marker for capillaries. So any transgene expression which overlapped with the lectin stain was termed endothelial selective, and any M-cherry expression which occurred outside the confines of the vessel was termed extravascular. So at the 0.1 megapascal pressure, we see nearly 95% endothelial selective transfection, and as we increase the pressure, we see an increasing extravascular fraction, which corresponds with the increased degree of blood-brain barrier disruption. Because we'd like to utilize this approach for stroke applications, we wanted to investigate the effects that these treatments were having on various inflammatory and immune response pathways. So we conducted RNA sequencing analysis 24 hours after treatment and found that at the higher pressures, 0.4 megapascals, which you can see in the image on the bottom right, is associated with very extensive blood-brain barrier disruption. We see a significant upregulation in a number of pathways related to inflammation and immune response. Meanwhile, at the 0.1 megapascal pressure, where we could not observe any blood-brain barrier disruption, we don't see any significant alteration in these pathways. And lastly, analysis of cavitation data has revealed a correlation between the extent of blood-brain barrier disruption, as measured by the contrast enhancement in T1 MRI, as well as the, between that and the microbubble emissions at harmonic and subharmonic frequencies, which can be used to establish a threshold between treatments which open the BBB, which is the pink portion of the graphs, and treatments which do not open the BBB, which is the green portions of those graphs. And this can be used to inform and monitor future treatments so that we can keep our treatments within a minimally inflammatory BBB maintaining pressure. So in conclusion, we've demonstrated that the endothelial selectivity of FUS-mediated transfection can be modulated according to the peak negative pressure of FUS pulsing. We've shown that endothelial transfection is possible independent of any detectable blood-brain barrier opening, and that this transfection does not appear to upregulate inflammatory or immune response pathways. And we've shown that acoustic emissions thresholds can be used to distinguish between treatments which open the blood-brain barrier and those which do not. So I'd like to thank the Price Lab and everyone at the University of Virginia who has supported this work, as well as our funding sources, and I'll take any questions.
Any questions?
Do you have any idea what the transfected cells are that are close to the vasculature?
Not yet. It's something I'd like to go in and stain for to figure out the identity of those cells.
Okay.